Hello, I am Professor Sims, and in this video I will continue discussing microbial genetics, specifically polymerase chain reaction, DNA fingerprinting, and Sanger dideoxy DNA sequencing. This is the last of the series of 10 lab sessions held as part of my Laboratory for the Fundamentals of Microbiology course. If you are a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and any other course information. Learning objectives for this unit include learning the concepts of PCR, DNA fingerprinting, and Sanger sequencing, knowing the difference between in vivo and in vitro DNA replication, learning the steps involved in performing PCR and DNA sequencing, and learning how to use the BLAST software to query your DNA sequences, and how to properly report and cite sequence data for your lab reports. That last note we'll talk about some more in the report video and also in class. Gel electrophoresis is used in DNA fingerprinting. It's a technique commonly used to separate biological molecules based on their size and charge. So this can be not only DNA, but RNA, proteins, other things. Now, as for DNA, due to its negatively charged sugar phosphate backbone, the DNA is strongly attracted to the positive electrode. So what you do is you cast a sheet of agarose, you put it in this chamber with buffer, and then you apply a voltage across it. So you have a negative charge up here, positive charge up here. You load your DNA up here, and the DNA is attracted to the positive charge. So in the agarose gel electrophoresis, the gel is oriented horizontally. In a buffer solution, the samples are loaded into the wells on one side of the gel, that's the side that's closest to the negative electrode, and the DNA is drawn through the molecular sieve of the agarose matrix down towards the positive electrode. The agarose matrix impedes the movement of larger molecules through the gel, whereas smaller molecules will pass through more readily. So the distance of migration is inversely correlated to size, with the smaller fragments traveling a longer distance through the gel and the larger fragments staying closer up towards the top. The sizes of DNA fragments within a sample can be estimated by comparing them to the fragments of a known size in a DNA ladder, which is also run on the same gel. So again, we'll go through this some more in class, but essentially what this ladder is, or sometimes it's called a marker, it's a piece of DNA that has been cut into much smaller pieces with restriction enzymes. This is done artificially, so they know exactly what size all of these fragments are. And you run that in the gel alongside your sample, and then you can use it to compare where, if you know what these sizes are, then you can determine what size these bands are. Most methods of DNA analysis require a large amount of DNA. The polymerase chain reaction, this is used to rapidly amplify DNA for use in other molecular analysis. PCR is an in vitro laboratory artificial technique, and it takes advantage of the natural processes of DNA replication. The heat-stable DNA polymerase enzymes used in PCR are derived from hyperthermophilic prokaryotes. The Taq DNA polymerase is derived from Thermus aquaticus. Thermus aquaticus, so you take the T from thermus and you add the AQ from aquaticus, and that's where you get Taq polymerase. Thermus aquaticus is isolated from hot springs. The first one was in Yellowstone National Park. The DNA replication requires the use of primers for initiation of replication, and they have to have a free 3' hydroxyl group available for the addition of nucleotides by the polymerase. So your DNA primers with known sequences targeting a specific known region of DNA are actually synthesized. It's done commercially, so you can actually send this off, tell them what you need, and they send it to you. It's amazing. These DNA primers bind to specific targets due to their complementary base pairs, and then the target sequence is amplified. So that's all very confusing. So let's just look at it step by step. In vivo DNA replication, separating the strands, unzipping the DNA, right? That's done with helicase. Then your topoisomerase is there to stabilize the sugar phosphate backbone, so it remains intact. In vivo, the primers, this is actually an RNA primer in vivo, and it's made by the enzyme Polymerase. Adding new nucleotides to the parent strand is done with DNA polymerases that are produced within the nucleus, right? Now, in vitro, unzipping of the strands is done using heat, and this is actually called denaturing. And what you do is you use a temperature that is hot enough to break the hydrogen bonds between the nucleotides in the center, but it's not hot enough to break the phosphodiester bonds holding the backbone together. The hydrogen bonds in the middle are much weaker than the phosphodiester bonds on the outside. So you find a nice sweet spot. You get it hot enough to where the hydrogen bonds break and open the strands up, but not so hot that the backbone breaks apart. Then your primers, like we discussed, they're artificially synthesized, and your polymerase, it's a DNA polymerase, but it's actually taken from a hydrophilic or a thermophilic Thermus aquaticus. It's taken from him. PCR occurs over multiple cycles, each containing 3 main steps: denaturing, annealing, and extension. So machines called thermal cyclers are used for PCR. These machines can be programmed to cycle through temperatures required at each step. They can cycle through these steps many, many times. So first, your double-stranded template DNA containing the target sequence is denatured at approximately, it's usually between 90 and 96 degrees Celsius. The high temperature required to physically separate the DNA strands is the reason that the heat-stable DNA polymerase, the Taq polymerase, is required. Next, the temperature is lowered to about 45 to 55 degrees Celsius. This actually says 48 to 72. I usually see it closer to about 55 degrees Celsius. But anyway, the temperature is lowered, and this allows the DNA primers to complementarily bind to the ends of the target sequence, and this is called annealing. So anneal basically means to stick. The primers stick to the template strands. The last step, the temperature is raised to it's usually about 72 degrees, which is the optimal temperature for the activity of the heat-stable DNA polymerase, the Taq polymerase. It allows for addition of nucleotides to the primer using the target as a template. So in English, this is what happens. In a tube, in a microcentrifuge tube, you make a little mixture, your reaction mixture, which is like a soup, right? You put in your soup, you put some water, you put some salt, and then all of this stuff here is your meat and potatoes or veggies. So you put deionized distilled water. Magnesium chloride is usually the salt that is used. For PCR, you're using an artificially synthesized primer, then your Taq polymerase, and this DNTP stands for deoxyribonucleic acid. That's your A's, T's, C's, and G's. And then you put that, mix it up real good, and you put it in your thermocycler to cook it, right? So it heats it up real hot, and it breaks those hydrogen bonds in the middle, and then primer comes and adds, and then that's when you drop the temperature, and then the Taq polymerase comes and adds new DNTPs. And this happens over and over again, usually like 20 to 40 cycles, and the machine cycles through all those temperatures automatically. Each cycle doubles the number of the double-stranded target DNA, and typically your PCR protocols, they have 20 to 40 cycles, and that allows for the amplification of a single target, single template DNA by tens of millions. So you start with a little bit and you end up with just tons of DNA. And this all happens within like about an hour and a half. It's really awesome. A basic sequencing technique is called the chain termination method, which is also known as Sanger Dideoxy DNA sequencing. It was developed by Frederick Sanger in 1972. This method involves DNA replication of a single-stranded template with the use of a DNA primer to initiate synthesis of a complementary strand, DNA polymerase, a mix of the 4 regular deoxynucleotide monomers, and a small portion of dideoxynucleotides, each labeled with a fluorescent tag or a flag, the DD NTPs. So they call deoxynucleotides DNTPs, dideoxynucleotides DD NTPs. The DD NTPs are missing a hydroxyl group at the site at which another nucleotide usually would attach, but it can't attach because the hydroxyl group is missing. Every time a DD NTP is randomly incorporated into the growing complementary strand, it terminates the process of DNA replication for that particular strand. And this results in multiple short strands of replicated DNA that are each terminated at a different point during replication. And when the reaction mixture is subjected to gel electrophoresis, the multiple newly replicated DNA strands form a ladder of differing sizes. Because the DD NTPs are labeled, each band on the gel reflects the size of the DNA strand when the DD NTP terminated their reaction. A dideoxynucleotide is very similar to a deoxynucleotide in structure, but missing hydroxyl group here, which is the key to this whole thing. So when a dideoxynucleotide is incorporated into a DNA strand, DNA synthesis stops because the next nucleotide doesn't have the little hydroxyl group to hook onto. The hook is missing, so it can't grab on. Again, we'll explain this just like for PCR. For Sanger sequencing, you make a reaction mixture, your soup, right? So your water is still there, your salt is still there, your primer, your polymerase, your DNTPs, and now you also add a small amount of DDNTPs, the dideoxynucleotide pairs. And then again, very similar to PCR, you put this into a machine that automatically cycles it through temperatures, and then it runs through a gel, and then there's a laser that can actually read which fluorescent tags are where. So as your reaction runs through this gel, the laser sees, for instance, red, and it knows that red is thymine. It sees a blue light, and it knows that that is for cytosine. So that is literally what is happening. As those dideoxynucleotides add, they stop their reaction and they glow. So depending on the size of the DNA that's synthesized, the laser can tell you exactly where that dideoxynucleotide added, where in the sequence it added, and because of what color it is, it can tell you which dideoxynucleotide added. You end up with this chromatogram that gives you not only the sequence, but it also can tell you, based on these peaks, how many times that specific DDNTP added to that position. The chromatogram shows where the DNTPs added, so the actual sequence itself, how many times each DDNTP added to that position, and of course where it is, which one it is, and how many times it added there. So the higher these peaks, the more reliable your sequence is, because that means that this C added to position 71, it's cut off, but probably at least 200 times. I'm going to run the sequences for you, and I'll post the sequences on Moodle, and then if you want to, you can go ahead and run your sequences through the BLAST database. If you want to wait, then that's fine too. We will do these together when we meet for class. If you can't stand the wait, or you just want to try it out, you can go ahead and do that. I'm going to run through the process with you here. So the first thing you need to know is what BLAST is. BLAST stands for Basic Local Alignment Search Tool. It's basically the Google of DNA sequencing. So what you do is you go to the BLAST website. You want to choose Nucleotide BLAST Program on the home page. You're going to copy and paste the sequence that I give you into the box labeled Enter Query Sequence. You're going to choose the nucleotide collection, the NRNT database. Where it asks for program selection, you're going to choose where it says to optimize for somewhat similar sequences, and it's called Blast In. And then you're going to click that blue Blast button on the bottom. You're going to choose the best match and report your findings. So the findings that you definitely have to report are your query coverage, your e-value, your percent identity, and the accession number. So this is what the website looks like. You're going to click this here where it says Nucleotide Blast. That will bring you to this page here. This is the box where you're going to paste your sequence. This here, you're going to select Nucleotide Collection, NRNT. Then you want to select Blast In down here, somewhat similar sequences, Blast In. And then you click the Blast button. That will bring you to a page that looks like this. It's important that y'all understand that this is a huge, enormous database where people have been adding sequences and primers and all kinds of information to this database since 1990. So it is enormous, which means it might take a few minutes for it to match your sequence to sequences that are already in the database. And you have this little splash screen here that will tell you how long it's going to be until the next update. So it might actually take a few minutes, and I don't want you to think that your computer froze or you broke something. It's totally normal. And then once it is finished searching, it will give you your results. The top of the page has a graphic summary. These little rectangles here kind of give you a visual cue of your query coverage, so that how many of the sequences matched something that was in the database. Each line going down is a hit or a match. And the first, sometimes the second hit is usually the best one. And the way that you can tell is if you scroll down the page, the next thing you can find are the descriptions down here. And these are in the same order as what you saw in the graphic summary here. So you're looking at the first two for this one because they had the most query coverage. And so for this one, the top hit, the best match is Klebsiella aerogenes. So it will tell you the description. If you click on this hyperlink here, it will take you to like more information, like the paper that first published it and how they discovered it and all that stuff. And the values that you want to report are all over here. So the percentage of query coverage, you want that to be 100%. Your e-value, if you have anything other than 0.0, it might not be a good match. For bacteria, you want your e-value to be less than 0.02. Your percent identification should be at least 99.5%. And then the accession number here, you absolutely have to make sure you report the accession number, because the accession number is like its identification number. Same way you have a social security number, all of these entries in the database have an accession number, and you have to report that. And I will talk to you some more in class and also in the report video some more about how to cite the BLAST database. There is a very specific full citation that you should use in your lab reports. So I want to make sure that we go over that. The Lab 10, we do not have a wet lab procedure in Lab 10. It's the last time that we meet before your project reports are due, before the final exam and all that. But we're going to do a lot. You need to make sure that you come to Lab 10. So what we're going to do is we're going to review the Lab 9, the DNA extraction results. We're going to review that all together. We're going to discuss the similarities and differences between in vivo DNA replication, in vitro DNA amplification, Sanger Dideoxy DNA sequencing. We're actually going to do an interactive group simulation. We're going to query the unknown sequences together on the BLAST database. And then we're also going to have an in-class review for the final exam. We're going to have Q&A for any kind of issues you're having with your unknown reports. So bring questions for me or for your instructor regarding all of the remaining reports, any material from any of the labs that you need clarification on. I will answer as many of those questions as possible before we run out of time. And finally, I want to thank you guys for watching. Don't forget to do the reading. Check the description down below for more videos related to these topics, and leave your questions for me in the comments below.